sent, like, instead of writing hey, I wrote he, and it just sent as he, and now it's just awkward. Okay, hey.
He.
So, yeah, so Lindsay, if you get a text, it just says he, so I'm sorry.
Okay. Got good.
Okay, there we go. That's a proper text message. Because, yeah, I can use words. Okay. Okay, there we go. I opened up the key 2 now, so we should be A-OKs.
Sweet. All right. I'll read question 1.
Sure.
It says, if the sequence of one strand of DNA is 3' T, G, C, T, C, 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 A, T, 5', what is the sequence of the complementary DNA strand? And I said that it would be 5' A, T, C, G, A, A, G, G, T, T, A, 3'.
Yep.
Yep.
Okay, that's pretty straightforward.
Yeah.
It's not. It'd be one thing if it was. I mean, it's replication, so it's going to be DNA, not RNA.
Mm-hmm.
Another DNA strand. Okay. Who wants to take 2?
I could take 2.
All right.
This question relates to question 1. How does knowing the structure of DNA help you hypothesize a possible model for DNA replication? Yeah, I think I kind of gave a little odd answer, but the answer to it is the DNA nucleotides in a double-stranded DNA molecule follow the ATGC base pairing rules, and the 2 DNA strands are antiparallel. As a result, a possible model for DNA replication involves reading each template DNA strand in the 3' to 5' direction, producing a daughter DNA strand with the opposite polarity and a complementary DNA sequence that follows that ATGC rules. So basically just a descriptive of what you just did. I guess that's what it sounds like.
Yeah. I don't know. Was that off the key?
Yeah, that's right off the key.
Yeah, it was off the key.
That was kind of vague to me, like, you know.
I wrote it like in the sense that I was like, if you want to know which type of structure it is, then using the complementary strand, you can compare that together, and then when it splits and stuff, and then when you keep replicating it, you can determine just from the first question in your complementary, like how far down it'll go, and then like what it'll look like towards the end. So like when you like in the random PowerPoint, there was like that photo of like when it like described all that random stuff, like of like each one. And so if we like just imagine those strands as like the colors, then I just said that we could just determine the end. It's just that my wording was really awkward, and so when I looked at the key in comparison to this, I was like, yes.
Yeah, I think mine was pretty like it explained what's going on, but I did it in like 2 sentences, so not very detailed.
Yeah, I did mine in 2 sentences too.
I don't know. I was like, I guess it doesn't matter. I guess I could just like read that and study that as the answer, you know.
I just
Go ahead.
Oh, sorry. I just interpreted it as like if you just understand like overall, if you just understand how the complementary works, and if you understand the structures, then I just said that. Then I just think that for number 2, if you just understand those kind of 2 ideas, and that the fact that complementary and the original strand are anti-parallel, then I just kind of went whatever. You understand it. Like that's. Because that's all it really is asking is like, okay, do you know what it looks like? And if you were given this strand, would you be able to determine, like, in the end, like, process? So.
Yeah. You want to do number 3?
Sure. What are the possible mechanisms of DNA replication? Describe how these three models differ from each other. Oh, shoot, sorry. Eileen just texted. She said, yeah, give me a couple of minutes, I'm almost ready. So she'll be here soon.
Okay. Ask her if she wants us to wait, because we just started.
Oops. Ah, crap. I am not tech savvy today. Question mark. There we go. Okay. Let's see what she says. In the meantime, should I answer 3 or should we wait? Oh, Agnes! Yay!
Hi.
Hi!
Maybe we should just wait a couple more minutes. We're
Okay.
in 3. So
Yeah.
did you have any questions with number 1 and 2 when you did the homework, Agnes?
So what happened is I turned in the wrong assignment.
Oh,
Oh.
Oh.
I turned in the applying genetics one because the day I did the assignment, I had 2 exams. I actually have an exam in a few minutes, but
Oh no.
Oh dear.
yeah. But yeah. So I just hurried to do the thing, and I wasted my time because I turned it in and she marked me as a 0, and I was like, what the heck? And then I emailed her and she was like, oh, I forgot to tell you that it was because you did the wrong assignment.
will she let you turn it in?
I asked her, but she hasn't replied.
She's pretty reasonable, I'm sure. I mean,
She didn't email me. She wrote it in the little thing, you know, that comment thing that comes.
Yeah.
So I need to ask her.
Yeah.
And see.
Okay. I'm sorry.
You must be at the Benson then.
yeah.
Yeah, I am, because I have
Oh.
a practical exam for anatomy physiology soon.
What time?
Well, the last time that they're letting us take it is at 4.
Okay.
But
Sure.
what do you call it? But because I haven't signed up, I'm trying to get into whichever one of the next. Because I don't want to go there at 4 and they're like, oh, we're full.
Well, maybe should we keep going then?
Probably something good.
Okay. I feel really bad now.
She
Oh,
text.
and then Eileen,
What did she say?
she said we can just go ahead. She'll be there shortly. Just send me the link.
Okay. All right.
Yeah. But we can keep going. So,
So go for it, girl.
okay. So this one is just asking about the differences between conservative, semi-conservative, and dispersive. I wrote it in a really sloppy paragraph, so I'm just gonna wing it and talk. But basically, I interpreted conservative English as like you start off with your parent and then splits off into 2, but instead of like an even amount, it's just one is exact copy of the first one and the other one is just a brand new like daughter strand like all together. And then from there it splits again, and then from the left side or like the blue side or parent side I should say, where it was like the exact replica, it would split off into again another complete new one, and then a brand new daughter, stepdaughter, daughter, granddaughter thing, granddaughter strand, and then from the on the other side, I'm having a bad day. On the other side, there will be the daughter, but then it will split off into 2 completely new stepdaughter, granddaughter things. So from there, it's just completely like I'm a copy and just keep going, and then. The semi-conservative is the one that we believe today and is the one that totally makes more sense. So I don't know why they didn't think of this, but in which case you start with a parent, and then you split off into 2, and then you imagine it as like each strand from the parent just split off altogether, and then from there a new strand interacts with them, and then so you have like a hybrid thing altogether, which is cool. And then from that one hybrid, which is the same again, and so the original there'll be one side of the parent strand that'll just have another new replica on it, so it'll just be together, and then it'll be a completely new one from the other strand that like split off, and then it'll be the same on the other side. And then the last one is dispersive, and dispersive in which you start with the parent again, and then you split off. You have 2 hybrids again of the same thing, but then instead instead of those two like kind of splitting, it'll be like each one is the exact kind of like. Correct me if I'm wrong, because this one
It was a little confusing for me.
I don't know what happened to me.
Yeah.
For me, how I thought of it as like it was like okay, so the first time it was like the parent, and then when it goes into the second area, it'll split, and then it'll have a new strand with it. But instead of splitting again, it just kind of replicates itself, and so it just kind because all four of them are hybrids in this case, instead of just like the semi-conservative in which there was two hybrids and two new ones. In this one, it's like all hybrids, and so the only way I would have seen it as all hybrids was if it was kind of like the conservative in which it was like it stayed the same, but then it like still replicated itself.
So the way that you're talking about dispersive, right?
Yeah.
So the way that
And the thing is, sorry, go.
the way that I understood dispersive is like, and you might I might not I might have
No, I
understood.
actually have no idea. So go.
But like, so there's the parent strand, and then whenever it replicates, new material is like within each. So within each, I'll show you a picture. So within each of the pink is the new material. So there's the old material, and the new material is throughout the strand. So in this one, there's a little bit of new material at the bottom. In this one, there's a little bit of new material at the top, but it also has old material in it. And then it replicates again. And at first, I thought that it still would have like new material and old material mixed in, and that could be the case. But the point is, in each replication, I think, because I'm pretty sure whenever in each generation, more and more new material gets mixed into, dispersed into the strand. So it becomes like, it becomes more pink. It becomes more like new, and more new is in the strands instead of old information. That's the way that I understood it. But does anybody else have any, like, knowledge about dispersive? No? It was kind of confusing. Hello? Hello? Can you guys hear me? Hello? Where did Shannon go? Hello? Hello? Can you hear me? Hello?
I can't hear you.
Can you see me?
Oh, now I can hear you and see you.
Okay, sweet. Okay. Well, I don't know where everybody else went. Oh, shit.
Oh, they're coming back. They're coming back. I'm just taking notes, so I don't know how to do this.
What did you hear about what did you hear that I said? Did I freeze or something?
Like
Yeah,
it
you
just went
did.
black.
Like in the
And
middle
then
of
it's like,
your.
Huh.
I don't know, but I just lost all internet connection, so I had to jump on.
Oh, okay.
me too.
No worries. But what all did you hear about dispersive?
I don't know anymore. I was just really lost.
Oh,
Like in the internet world, not in the text.
Yeah.
this is how I understood it. Like there's new material. I mean, there's old material, so there's the printle, and then whenever it replicates, they thought that new material kind of showed up. So like in this one, the pink is the new material, so there's a little bit of new material up top here, and there's a little bit of new material in the bottom here. So it's just showing that it's dispersed within the old material, like it's mixed together. And then as it replicates, more and more it becomes new. That's what I understood. Is that how you guys understood it?
That makes a lot more sense because the name would then make a lot more sense.
Well, does it ever become completely new? Like, does it ever become all new, or is it always going to have some of that mixed stuff, mixed material?
It says in the key that after one round of replication, the dispersive model predicts that both DNA strands in each daughter molecule will contain original and newly synthesized. So I think it would be kind of like what you said, in which it's like the old and the new, but instead of just a completely new daughter, I think it would still be like a little bit of old, a little bit of new. I think that's what I'm getting from it. If not, then I'm just an incomplete. But
it has the thing that it has N14. It has a band up near N14. Hold on, let me look.
wait, what about N14? I didn't hear that.
Or maybe a single band located at the half. That's 4, not. Are we on number 4? We're on number 3.
I mean, number 4 deals with conservative and all that stuff.
Oh.
Replication, the original DNA double helix fragments into pieces. These pieces are then distributed to the daughter DNA. Any gaps are filled in with newly synthesized DNA. After one round of replication, the dispersive model predicts that both DNA strands in each daughter molecule contain original nucleotides and newly synthesized nucleotides, whereas the semi-conservative model predicts that each daughter molecule will contain one original strand and one newly synthesized strand. So yeah, dispersive will have like a mixture. But I'm trying to figure out Okay. Yeah, anyways. So that's right. I mean, it's mixed instead of having one old strand, one new strand. Like semi-conservative. Okay. Who wants to do 4?
I can do number 4 since I just came into this.
No worries.
Okay, so the Michelson-Stahl experiment was based on the ability to separate heavy N15, half heavy N15 and 14, and light DNA N14 in an ultracentrifuge. What would be the result after a single generation of DNA replication? Result after a single generation if DNA replication uses the conservative, semi-conservative, and dispersive models. So these are really similar to number 3, but like, let me see the answers now. I haven't looked at these yet. So on the answer key, it says that for the conservative, 2 bands of equal intensity would be observed in the test tube. One band would be at the heavy position, and the other would be located at the light position. For semi-conservative, it would be 1 band at the half heavy position in the test tube, and then for dispersive, it would be one band also at the half heavy position.
That makes sense.
Okay. Let's see, how many parts is this?
And what about the second generation? Because that's where, like, I understand the first generation because, like, conservative, it has the parent with the old information, and then it has the daughter with all new information. So it's going to be equal. Like, I get that. And then semi-conservative, it makes strands with half old, half new. So it would be mixed. So that's why it would be in the middle.
Mhm.
And then dispersive, it's all mixed throughout the old strand, new strand. New information is mixed throughout the old information. So that would be mixed too. But it's the second generation that I start to get like more iffy about.
So for the second generation, it says it for conservative, it says 2 bands would be observed in the test tube again. So one would be at the heavy position, and it represents 1/4 of the molecules in the reaction, and an intense band would be located at the light position, and it would represent 3/4 of the molecules in the reaction. And then semi-conservative, 2 bands of equal intensity. One would be at the half heavy position and the other would be at the light position. Dispersive is a single band located halfway between the half heavy and the light position.
I guess semi-conservative is where I get confused. Because, like, is there a picture of, I haven't looked at the PowerPoint yet. Is there a picture of, like,
There's a picture of what it looks like the half heavy and heavy.
is there,
Of that? No.
no?
I don't. No, the most that like picture-wise it gives, it's like the little like strandy things like that.
Yeah, it doesn't really tell you much. That's how I thought.
Yeah.
Because, okay, so, well, I'm probably getting on y'all's nerves, but I'm just trying to figure out. So, with semi-conservative, there's old information, and then whenever it splits off, when it replicates, then there's one strand or one DNA molecule that has the template strand still, one parental strand, and one new strand. And then it has another DNA molecule with one parental and one new strand, right? So that's the first generation. And that would make sense because it said the first generation was mixed. So it's in the middle. But the second generation, I would think that, oh, okay, never mind, I'm stupid. Okay, so the second generation, it would have, so the parent is now the parent molecule now has one strand that's old, one strand that's new. So the daughter will have one strand that's old, one strand that's new, and then 2 strands.
So like,
Right?
girl, I mean, with that.
And then what?
I don't know if you are.
So, okay, I originally thought that it was like this. I don't know if it makes sense to you, but so like, there's a parent, semi-conservative method believes that, there will be old, new, old, new. And I originally thought that it'd do the same thing and it'd be old, new, but it uses each of these as a template strand. So it would be old, new, and then new, new.
Yeah.
Here.
Yeah, it is new.
That's why there's more light. It makes sense now. I just wasn't getting it. Sorry.
It's all good.
Yeah, that makes sense. Okay, you guys can go ahead. Sorry.
Uh,
No, you're good. No, you're good.
okay.
Dot.
Okay.
Number 5, guys.
Five.
Okay, so 5 is a drawing thing. So how do we want to do this?
Hmm.
You want to just talk it out, I guess?
Sure.
I don't know.
No, because I have no idea. And like the key doesn't have anything either. It's just I will provide a drawing.
Yeah.
Yes, you can leave whenever you need to leave. Like, seriously. I feel so bad that you have a test at 4. Like,
I'll leave in like a few more minutes just because I think this is because I still have to do this assignment,
okay.
so this is helping.
Okay.
I was a little bit confused by this one because I get a draw linear double-stranded DNA molecule indicate. Like we all know that it's polar, and the origin of replication is in the middle of each strand, right? Like that part I get. But then, how did you guys draw the early versus later stages?
I think the early is like when it first starts, and then like the later one, I thought it was like that weird circly thing with like the Ozaki fragment, but I'm really bad at talking.
But what does the early stage look like then? Because that's what I thought that all the stages, like I don't know.
think maybe on either slide 7 or 9.
I need to pull that up.
It looks like they're just barely starting the whole splitting apart of the DNA.
Yeah. So, and then is the
Probably
on
7.
slide 7 like the little and then on 7 like the little circle thing like when it's like all together. I guess that would be the one when it's doing the later stage, because the beginning would be like, oh look, I'm splitting, rawr. And then later on it's like, look, I'm a circle, I'm gonna eat things. Just kidding, it's not eating, but I'm gonna stop.
So basically, I need to look at. I haven't looked at the PowerPoints. I'm always really bad at that. PowerPoints just don't like. They're not my thing. But
That's okay.
I was the opposite. I would only look at the PowerPoint and the notes, but I would never look at the videos. And so anytime it's like, look at the video, I'm like, shoot.
And I like the video, but I don't like the PowerPoint, but I don't like the notes because I just don't like doing all that reading. Like it just takes so long.
There is a lot of words in there. I hate it when it's like 21 pages or something.
Yeah, I'd rather just like look at it and have it be visual. It makes more sense.
I'm able
Yeah.
to
I'm tired.
okay.
The sleepiness is real. I just want to take a nap.
I'm allowed to look at like written stuff at work, and that's the only time I have to study for this class. So I tend to watch the videos or look at the PowerPoints because I can print out the notes and have time to read those, but everything else is kind of harder.
Yeah.
But anyways,
Sorry.
that's okay. Life. Who wants to do 6?
Okay, cool. If someone can explain 6, that would be wonderful, because I am not the most graceful with this.
I kind of can.
Okay.
So in 6,
I'm gonna go now, girls,
okay.
Oh, okay.
before you get started on that one.
Good luck.
Thanks. See ya.
Bye.
Yeah.
Good luck.
Thank you.
So, an initiation, and this is for like E. coli, right?
Yes.
It just says prokaryotic, so yeah.
Yeah, we can just say E. coli.
Okay, so an initiation, DNA. I wish I need to look at the PowerPoint because I'm sure it has something, but pictures help me more with like understanding. But the DNA binds to DNA boxes and uses ATP to separate the strands, and then helicase uses ATP to form replication forks. And then the single-stranded DNA binding proteins prevent the strands that have separated from coming back together and forming hydrogen bonds. And then elongation starts, and I don't know, it kind of seems like the same thing to me, but DNA helicase, so it's formed those replication forks, and the strands are separated. The single, that's all part of the initiation, but I wrote it again. DNA gyrase introduces negative supercoiling that makes up for the positive supercoiling that helicase did. DNA primase synthesizes DNA primers. It consumes ATP. And DNA polymerase III synthesizes the leading and lagging strands at the same time, and it consumes the DNTPs. And it also proofreads. And then DNA polymerase I removes the RNA primers that are that make the Okazaki fragments, and it fills those primer gaps with DNA, and it also proofreads as well. And then DNA ligase connects all the Okazaki fragments together, because even though it's filled in, the polymerase I fills it in with DNA, there's still like teeny tiny like spaces in between each. And so the ligase kind of smooths it all out and connects it. And then termination, I think this is right. In termination, 2 displaces replisomes and terminates replication. DNA ligase forms final covalent bond in the newly synthesized DNA, and DNA gyrase resolves any intertwined chromosomes, because sometimes, so like bacteria is in a ring, so sometimes together and it separates them into 2. That's what I got.
The only thing I was really confused about is that, like, when it got from, like, 6A and B, when it talked the ones that consume ATP or the ones that consume DNTPs, anytime I would, like, type in the word, like, consume, like some would show up, but none of them matched the answer key, and so I was like, shoot. And the only one I got for, like, any of them was, like, the DNA helicase, and that was only for 6A. Everything else I got, like, completely off chart, except for, actually I got DNA polymerase III, but still, oh, and DNA polymerase I, I got that too, Oot. But I didn't get the holoenzyme, and I didn't get the DNA or DNA gyrase or primase or ligase, because I didn't read in that, or I guess I didn't understand that it would consume it, so.
What did the key, I keep forgetting that there's a key. So what did the key say about this?
It just said a whole bunch of proteins that did consume it, but what I was saying is that I didn't get any of it except for the DNA helicase.
Okay.
Like in the instructor notes, it says like DNA ligase consumes ATP, DNA polymerase I consumes the like DATP.
Oh wait, I took it like
ATP, blah blah blah. So like each step, it like says which one that they're consuming.
Oh wait. Oh crap. I just keyed my own paper wrong. Cool. Okay, hold on. Oh, whoops.
This is gonna
Okay.
remember for the test. All these little details.
No, for real.
Yeah.
Remembering all this is gonna be fun. Woo. Okay. JK. The only thing I didn't get was DNA and holoenzyme. Where is holoenzyme?
That is I think it's polymerase III. I can't remember what it's made up of. It's
Alright,
like a,
polymerase 3 is holoenzyme?
yeah, DNA polymerase 3 is a whole enzyme. So whole is not, enzyme is an enzyme made up of different parts. I don't remember. Let's see if I can find it in here.
Okay, JK. I got it. Cool. I did not know that fact. Okay. Cool. Sorry guys.
Good. Oh, it's made up of subunits like alpha, beta, gamma, epsilon, tau, and theta. And each of those performs a certain function.
Okay. I'm going to hate this test. Okay.
Yep. Who wants to do 7?
Last question.
Let me pull it up really fast. Okay, so name and describe the functions of the enzymes involved in the 3 phases of eukaryotic DNA replication. Okay. So for initiation, the origin recognition complex. I'm just going to kind of read through what the answer key has because that's going to be the easiest way to do this. So it binds to each ARS element, and then regulatory proteins, CD, C6, and CD21 bind to the ORC and inhibit the initiation of DNA replication. And then MCM helicase. Let's see. It says it's a multi-subunit protein complex that catalyzes DNA strand separation, forming 2 bidirectional replication forks. It just makes the replication forks. And then in elongation, MCM helicase again. So it keeps forming the replication forks. DNA primase and DNA polymerase alpha. So, okay, these 2 things make up the one protein complex. And then as a DNA primase forms the first 10-ish nucleotides of the primer. And then DNA polymerase alpha forms the remaining 20 to 30 of the primer, nucleotides of the primer. And then DNA polymerase epsilon synthesizes the leading DNA strand at each replication fork. And it has a 5' to 3' polymerase activity. It also has proofreading activity. And then DNA polymerase delta synthesizes the lagging strand at each replication fork, and it also proofreads. And then pCNA, so it's associated with DNA polymerases epsilon and delta, and it clamps the enzymes to each template DNA strand. It increases the processivity, I can't even read that word, of DNA polymerases epsilon and delta. And then for termination, it just says there's no known protein factors involved in the termination stage of DNA replication in eukaryotes.
Sweet.
Yeah.
I got that totally wrong. For some reason I couldn't find that, but we'll have to study that again.
Is it not in the notes?
I didn't find it easily in the notes, but if someone else did, I would like to know where it is.
Yeah, I had a hard time finding all that. Did anybody find that in the notes?
I didn't, I found a few things for number 6, for number 7, but I didn't find it all,
here it is.
or where it's supposed to be at.
Because what I had a hard time is distinguishing what was for eukaryotes and what was for prokaryotes.
Yeah.
It really, I felt like it skipped around a little bit, but now I see where it says replication initiation, replication elongation. That's talking about eukaryotes. I don't know what page. I just found it.
Oh,
But
yeah, I see it.
It wasn't very clear as far as I mean, it says origins in eukaryotes, so I guess we should have guessed then. But I don't know.
Yeah, it goes through like each step.
Yeah. So my bad.
It's okay.
I just didn't like the setup because I was like, where is it? Oh wait, it's right in my face. Just kidding.
Okay, well, anybody have? I mean, I think me personally, like, I'm gonna go through and study again. Plan on doing that this Saturday.
I feel like a
What?
Uh,
Shit in your face right now.
no, I was just super confused. She just said she needed to study more this Saturday, and I was just about to say that a lot of this I feel like is like a terminology thing, and
Yeah.
it's like not really like unless we understand our own learning style. It's one of those things where it's like we can talk it out, and hopefully we'll understand. Like how Lindsay helped me with the dispersive model. But other than that, it's not like one of the math problems where we can be like, it was this number. How'd you get that number? Oh, we did it this way. So,
Exactly.
Yeah.
But if we have any questions, we can like text and stuff.
True.
Okay.
Sounds good. I'm gonna go take a nap, you guys. I'm so
Okay.
tired. I can't stay awake anymore.
Well, have a good night, and I'll see you guys probably next Friday.
Okay.
Yep, probably.
Probably.
Okay.
I'll send
Yeah.
another text to make sure that we're doing Friday. So.
Okay.
Okay.
Thank you.
Mm-hmm.
Cool. You're welcome. Okay, bye.
Bye.
Awkward buys.